Better turn off my phone. Yes. I can't go to the bathroom. They had a great scene in that Leslie Nielsen movie where he's mic'd and he goes. Speaker's nightmare.
Yeah, exactly.
Yep.
I'm Michael Derived, Director of the Institute for the Arts and Humanities, and welcome to this fall's final installment in our two-year project, first year of the two-year project, The Boundaries of the Human in the Age of the Life Sciences. And you will find out, I think, over the next hour why we call it The Boundaries of the Human, and I think the life sciences part explains itself. Our guest today excuse me, is Professor Scott Gilbert. He is the Howard A. Schneiderman Professor of Biology at Swarthmore, where he teaches developmental genetics, embryology, and the history and critiques of biology. He received his BA in both biology and religion from Wesleyan University, thereby bridging the two cultures divide all by himself. And he earned his PhD in biology from Johns Hopkins, working specifically in the pediatric genetics laboratory of Dr. Barbara Midgen. His master's degree in the history of science, also from Johns Hopkins, was completed under the supervision of Donna Haraway. Gilbert now currently has 3 books in print: Developmental Biology, a textbook in its 10th edition; Bioethics in the New Embryology, a volume co-authored with 2 students that discusses new findings in developmental biology with respect to philosophy and religion; and Ecological Developmental Biology, a textbook co-authored with David Eiffel, which integrates developmental plasticity, epigenetics, and symbiosis into discussions of medicine and evolution. Okay, just to catch you up on where we are in the Boundaries of the Human series, in September, Agustin Fuentes came from Notre Dame and argued that we need to rethink the parameters of our species at the interface of contemporary evolutionary theory, emerging data on physiological and epigenetic systems, and human social complexity. Then in October, Hannah Lendecker came from UCLA and gave us a history of biology and a biology of history, arguing that ideas about metabolism have undergirded cultural assumptions and practices about ideas of interiority, and how they undergird notions of the individual to the interrelation between animals and plants. Today, Professor Gilbert will suggest to us that if we take these arguments seriously, and if you haven't seen them yet, they're all on our website. We have the lectures videotaped on the Boundaries of the Human website at the Institute for the Arts and Humanities. If you take those arguments seriously, we will come to realize, Professor Gilbert will argue, that we are not individuals at all, and that symbiosis, once relegated to the margins of evolutionary theory, is in fact what makes the circle of life go round. please join me in welcoming Professor Scott Gilbert.
Well, thank you very much. I have a slight frog in my throat, which I hope won't hatch. It's been a lot of fun being here, and thank you so much for the invitation. Thank you so much for the hospitality. It's just been wonderful. I really have enjoyed speaking with faculty and students here enormously. I'll be talking today, but actually I'm going to just say coming to the campus and seeing the Waring buildings and the Waring, and then some of the people didn't know about Fred Waring, and Fred Waring, musician, alum of this college, and the inventor of the Waring blender, which used to be in every American household. Okay, this was the apparatus that started molecular biology. If you don't know about the Pajamo experiment, Pardee-Jacob-Monod experiment in the Pasteur Institute using the Waring blender, it's a beautiful experiment, one of those elegant experiments which basically mapped the E. coli chromosome using very little than a Waring blender. Anyway, I'm going to be talking today, I'm going to try to say that we are all lichens. It used to be that lichens were the emblem of symbiosis, that symbiosis was a marginal field. It was not mainstream. Recent studies have basically shown that symbiosis really is the signature of life on this planet, and that we have to talk about organisms as more than just these monogenomic individuals that come from the fertilized egg, but also as assemblages, multi-lineage organisms that act together as teams. So I'm going to start off by talking about social issues, actually. I'm going to start talking about kind of society and then come back through the body to society again. I'll talk just briefly about the body politic, this metaphor wherein a nation is considered to be a corporate entity, a corpus, a body. And what's true for the body is true for society, and what's true for society is true for the body. And we have things like Durkheim's notion, division of labor. Well, that's from physiology. The organ systems of a multicellular body show compartmentalization of function, and they work together towards a common end. Durkheim, of course, division of labor is key to regulating modern societies. Each person contributes to the functioning of the body by the individual tasks. This also gives them a shared goal and a consensus, which helps keep social order. This notion of body politic is very important. But I'll say that there are many different definitions of the body. And this is something that really is not very well understood, that what scientists talk about the body can be different from science to science to science. So there's the body politic that's defined by the immune system. In other words, can I defend my turf? Boundaries are defended by an immune network. If I were to put my skin on yours, you would reject it. Then there's the body politic defined by the nervous system, culture, Western civilization, the American way. If I were to put my brain into you, you'd be me. The brain is what defines, not the immune system. And then there's the body politic defined by the genes. Actually comes from the word gens, the tribe. My genes determine who and what I am. I would risk my life for those who I share ancestry with. Kind of, this inclusive fitness notion. So there are different bodies and different body politics, and one can extend this into bodies of knowledge. Bodies of knowledge defined by immune system, nervous system, genes, and so forth. But I just wanted to mention that these are different ways, and this means also that there are different diseases of the body politic. So we've gone through 3 of them, actually, in my lifetime. Infection, where the body is inherently pure, but susceptible to outside influences. communist infiltration, the quarantining of Cuba. The body's pure but susceptible to the noxious influences from the outside. That was one metaphor. There's cancer. The disease is from part of the body that has a function but which is unregulated. It's escape regulation. Media, the military-industrial complex, cancer on the presidency. We have met the enemy and they are us. Okay? The disease is from active internal agents. And then there's decrepitude and degeneration. The infrastructure is falling. Disease from old age. And we can easily see that when we're talking about the body and the body politic, the anxieties from one easily meld into the other. Now, what I'm going to say here is that when we talk about the body politic, we're using the wrong body. That this isn't the body anymore. The body has changed. And this is where the science of symbiosis is deconstructing the old notion of what an individual body is, replacing it with a new notion of individuality. We had been looking at a view of the body which was genomically pure, a body where each cell is genetically similar, shares a common embryological history. But that is not the body that I think we're going to be working with. When we say that a body is an individual, we usually characterize it in 6 ways. There's the anatomical individuality. You look around you, and you see different individuals. We've separated ourselves from the environment. There's physiological individuality, the integrated organ systems to a common end, what Hannah Landecker talks about. There's developmental individuality. This is something Thomas Huxley proposed, that an individual is the product of the fertilized egg. Our society, if you will, has a common origin. There's immune individuality. Immunology was described as the science of self versus non-self discrimination. We reject others. Very strong, actually, during the 1980s, during the AIDS epidemic, that our immune system exists to protect us from the environment. Genetic individuality. The genome of every cell comes from the sperm-egg fusion and is nearly identical. That we are genetically individuals. And then there's evolutionary individuality. Individuals are those that compete in the contest of natural selection. So these are the ways we've been thinking about individuals. But if you think about animals, what you find is that they're not really individuals. Think of the cow. Eating grass, one end, probably producing methane at the other end. Okay, the cow is eating grass. Only the cow can't digest grass. The cow's genome does not make enzymes allowing it to digest grass. The digestion is occurring in its rumen, in this huge fermentation tank that it has in its belly, and it's the symbionts that are digesting it. Similarly, the termite Mastotermes darwinensis, which is kind of a poster organism for symbiosis, it's an agricultural pest. It eats wood. It eats forests. It eats trees. It eats homes. Only it can't. Its genome also has nothing which allows it to digest lignans, nothing that allows it to digest cellulose. There's an organism in its digestive tract called Mixotricha paradoxica. And Mixotricha does the digestion of wood. Only it doesn't. Mixotricha is a composite organism of a protist and 4 types of bacteria. Together, all of them will make an organism. And actually, you could even say, is this really an organism? It can't reproduce. It needs a hive. So individuality is called into question by the termite at all levels. And then the coral over here, which I'm glad to speak to Monica Medina's laboratory today. The coral is not an organism without its zooxanthellae symbionts. It needs the symbionts for carbon and oxygen resources. And the bleaching, which is going on at an unprecedented rate right now in the world, is causing the symbionts to leave the coral, and the coral dies. It needs the symbionts. So I want to talk about these levels of individuality, this notion of what it is to be an individual. What we call this is the holobiont, the whole organism, the animal or plant, plus its persistent microbial communities. That the cow can't exist with its own genome, its own cells. It needs the symbionts. The coral can't exist without its symbionts. The termite can't exist without its symbionts. Organisms don't exist without help from their friends. So, anatomical individuality. This is a cross-section of the hive where I grew up. This is 54th and 5th Avenue, New York City. And you can see the different individuals here. What I like about this print, it's by Kitaoka. This used to be the Mutual Benefit Life building. And that's what this is all about, mutual benefit life. The individual, I'll take this individual over here. We are a collection of ecosystems. We are a walking assortment of different ecosystems. The mouth ecosystem differs from the air ecosystem, differs from the airways ecosystem, skin, intestines, all the pores on our body. Left hand differs from the right hand. We use them differently. All different ecosystems. Over 50% of the cells in our body, anatomically, 50% of the cells, probably close to 90%, are prokaryotic. They're microbes. Most of the cells in our body are microbial. Specific locations, about 160 species per person, about 1,100 major species in humans. It's been called the 11th organ system. And as I said, we are niches for bacteria. That's how we're seen in the world. And remember, as Stephen Jay Gould said, we talk about the age of the fish, the Devonian, and we talk about the age of the reptiles and the age of the mammals and now the Anthropocene, the age of the humans. It's the age of bacteria. Always was, always will be. Okay. Physiological individuality. So we're not anatomical. 50 to 90% of our cells in our body anatomically are microbes. Physiologically, this is this notion, Hobbes' Leviathan here. Henri Milne Edwards, Rudolf Leuckert were some of the biologists who were proposing that the organism is an individual whose component parts cooperate for the benefit of the whole. It's true. But it's not only our cells, it's the microbes. And here is a picture. This is Planococcus, which is a milkweed bug. It's a true bug. Planococcus, the insect, has in its cells Tremblia, a bacterium. And Tremblia, the bacterium, has within its cells Morinella, a smaller bacterium. Now, to make phenylalanine, to make the amino acid phenylalanine, the pathway actually starts in the bacterium, and then it goes into the bacteria's symbiont. Then it goes back into the bacteria, and the final step is done by the enzyme encoded by the genome of the insect. So in order to get phenylalanine for protein synthesis, it's a three-way symbiosis here. And that also is found in mammals. The microbiota, especially those in the gut, are making short-chain fatty acids. They're making AMPs. They're making tripeptides, which are keeping the circulatory system, the digestive system, the immune system, neuroendocrine system, and the bones in register. It's a coordination system for the body. So the microbes are actually helping our function. And what we found in germ-free mice and in mice where you could take out particular microbes, these microbes are incredibly important in the normal day-to-day functioning of the body. How do they do this? They will actually metabolize for us. So the food, dietary tryptophan, what's found in our cheese and turkey, what makes us tired in Thanksgiving, that gets transformed into indole in the gut. The gut can take it to various forms. It can send to the liver. And what you find is that there are different compounds which are made in the body only because the bacteria are present. For instance, serotonin. Serotonin not in the brain, but in the body, is actually produced through the bacteria, through tryptophan metabolism. Germ-free mice have hardly any serotonin. 95% of the serotonin in our body comes from the bacterial metabolism. Pregnancy is actually mediated in part through microbes. This is some work by Koren et al. In the Rosenberg lab in Tel Aviv. What they found is that the bacteria of a woman's gut and reproductive tract change during third trimester. It changes towards the end of pregnancy. The woman is getting a different set of bacteria in her reproductive tract and in the gut. And this bacteria, if you take it and put it into a germ-free mouse, actually causes some of the symptoms of pregnancy. You get a mouse with altered fat metabolism and insulin desensitization. You get a different type of bacteria here than you would during first or second trimester, which doesn't change the mouse's phenotype much. So during third trimester, you get inflammatory cytokines, you get different energy metabolism, and the gut microbes are changing. This is actually important. So pregnancy, in part, changes the bacteria. The bacteria, in part, help change the symptoms of pregnancy. The infant gets its bacteria from the female reproductive tract and from the distal part of the digestive tract. As the amnion breaks, the water sac breaks, the fetus is open for colonization. John Donne said no man is an island. Sociologically, he was right. Biologically, he was wrong. That's exactly what we are. We are islands open for colonization, and the rules of island biogeography hold. The first colonizers are critical for making the rules for the next set of colonizers and so forth. So, the mother has changed the bacteria in her reproductive tract and now is going to do something that didn't make any sense until this notion of symbiosis came about. The mother feeds the bacteria. There are certain saccharides in mother's milk that cannot be digested by any mammal. And they're not for the mammals. They are for bifidobacteria. Bifidobacteria has 7 genes in a row which are involved in metabolizing these sugars. It's good to have bifidobacteria in your gut. It's great for bifido to be one of the first colonizers and to have it spread throughout the gut. And what is the mother's milk doing? It's not only giving nutrients to the baby, it's giving nutrients to bifidobacteria, saying to the bifido, here are the supplements for your growth. Hardly any other bacteria can use this, and the mammals definitely can't use it. It's for you. And so basically, the mother is giving a new set of bacteria to the infant and now causing certain of those bacteria to grow more than others. So birth is not only the birth of an individual, it's the continuation of community. This mother's community of microbes is another inheritance that's being passed on to the infant. So we are not anatomically individuals. We're not physiological individuals. 30% of the metabolites in our blood come from microbial metabolism. It's like another liver. And indeed, the gut biome is equivalent in metabolic activity to the liver. So we're making a lot of material from our gut microbes. And so when we say that the fetus, for instance, is in an aseptic environment, it doesn't mean that it's not being influenced by microbes. Microbes are going right through the placenta, the microbial products. Well, what about genetic individuality? This is the big one. This is the big one. When we talk about individuality in America, we are usually talking about genetic individuality, the notion that all the cells of the body are the same. This is what's taught to us, genetic personhood. Each of us is a polity that consists of those cells that descend from the fertilized egg. Each of us is a genetically unique individual. Here's an ad that I heard on television a week ago revealing what it is that makes you, you. That's DNA. Send us your DNA. We'll tell you what makes you, you. Okay. Stand up, girl. You see this quotation in a lot of anti-abortion websites. Even more amazingly, intelligence and personality, the way you look and feel, were already in place in your genetic code. At the moment of conception, you were essentially and uniquely you. Susan Lindy and Dorothy Nelkin published this very interesting book, The DNA Mystique, where they looked at the genetic notion of personhood and came, they didn't read PNAS and Science and Nature. They read Red Book, Cosmopolitan, Newsweek, U.S. News and World Report, New York Times. And what they found of the rhetoric of DNA is that DNA was being discussed as soul. That which is your essence, that which determines your behaviors, and also that from which you could be resurrected a la Jurassic Park. My wife's background is Finnish. She got a newsletter that said, the sauna is in the DNA of every Finn. Well, Svante Paavo has sequenced a lot of Finnish DNA. No, it is not in the DNA. It's in the soul of every Finn. But this notion of DNA as the secular equivalent of soul, that which is your essence, is being told to us daily. Okay. The mid-sized Hummer, same DNA, smaller chromosomes. The essence is the same, the package is smaller. Jeep Compass, it's in the genes. The new 2011 Jeep Compass is genetically engineered. No, I hope it's engineered, but genetically engineered. Okay, legendary bloodline. The Sportage is genetically modified, armed with completely modified DNA. Okay, we're being told day after day after day that DNA is our essence. It's hard to get away from this. And you get this syllogism here. DNA is our soul, given we get DNA at fertilization. Therefore, we receive our soul and become individuals at fertilization. You see this in the Republican media all the time. Here's Huckabee on the Daily Show. Pro-life is scientific. Biologically, life begins at conception. That's irrefutable from a biological standpoint. Rubio has been saying this just about all the Republicans. But actually, what we do know, and I could say with absolute certainty, you're hearing it. Scientists don't know when life begins. That there's a debate among scientists as to when life begins. There is no certainty. Whereas certain political people will say, all scientists say that life begins at fertilization. No, there's people who say it's at fertilization, people who say it's at gastrulation. You can't form twins after gastrulation. Before gastrulation, you can form twins. So you can have the same DNA in different people. Some people say that it's when you get your brain waves. If we're willing to say that death is the loss of the brain wave, how about human life being the acquisition of the EEG pattern? And some people say it's at birth. So there is a debate here. But we're being told constantly it's DNA, it's DNA, it's DNA. Okay. From the holobiont perspective, we are not genetic individuals. We get a transmission, an inheritance of symbionts. In many insects and invertebrates, it's from the germline. If I were to ask my developmental bio students, what is this a picture of? I hope they would say, oh, this is a germarium of a Drosophila or some other insects, and what we see here are the nurse cells, and they are metabolically active, and they are providing material, this red stuff, and they're putting it into this cell, which is going to be the oocyte. And they'd be absolutely right. That's what this is a picture of. But it's not bicoid mRNA, it's not ribosomes, it's not mitochondria. This is Wolbachia symbionts. These are bacteria which are being transported into the oocyte. And that's how Drosophila gets its inheritance, just as it would mitochondria. It's getting it from the germline. We get our symbionts by infection. As soon as the amnion breaks, we get symbionts. And then we get them from mother's skin, we get them from Big Brother, we get them from the dog, we get them from wherever bacteria are. Now, the bacteria in our body, 8 million genes, compared to a measly 22,000 from the human genome. So we're getting a lot of genes from the bacteria. We don't know if these are active in humans. We do know that they're active in insects, such that this is an example of a genetic change in the bacteria causing a phenotypic change in the holobiont, in the whole organism. This is thermotolerance in the pea aphid. Now, the pea aphid is parthenogenetic. No males in this species. Female to female to female to female. It modifies its meiosis so that you always end up with females. Okay. Now, you could have different bacteria, though, in these females. One type of bacteria, has the normal wild-type allele, which makes a chaperonin protein, a heat shock protein. The organisms with this full allele, shown in the dark pink, they give rise to a certain number of offspring, no matter what the temperature is. They're thermotolerant. They make slightly less offspring than those that are not thermotolerant during most of the year. But when the temperatures get high, those who have the bacteria without the thermotolerance allele, who have a mutant thermotolerance allele, they can't produce offspring at all. So you have variation here. You have some aphids which do better at low temperatures, some that do better at high temperatures. This is not due to anything in the aphid's genome. It has to do with the genome of the symbiont. So here's something that the holobiont is, the phenotype is in the holobiont, but the gene actually is coming from the bacteria. This is a similar thing, again, with pea aphids. Without Rickettsia bacteria, red aphids become red adults. However, if you infect with Rickettsia, if you give them the symbiont, the red aphids become green adults. Depends if you want to be on the stem, if you want to be in the leaf litter. This is a selectable trait. So, but what's being selected is not the genome of the aphid itself, it's whether or not it has Rickettsia. Humans do have differences in their bacteria. One of the most interesting comes from Japan. The Japanese version of Bacteroides plebeius. Bacteroides is a wonderful bacteria in our guts. The Japanese version of Bacteroides plebeius has two genes, not found in the American or European versions of Bacteroides plebeius. These two genes come from horizontal transmission from a Bacteroides that lives on algae. These genes allow the bacteria to utilize the sugars found in sushi. And so it hasn't been shown that the people get advantage of this. But certainly there are different strains of bacteria in different populations of humans, and whether or not they contribute to phenotypic differences is something that's being studied. Okay, so we're not individuals anatomically, we're not individuals physiologically, we're not individuals genetically, we're not individuals developmentally. Immunologically. Okay, immunologically. When I was teaching immunology in the 1980s, basically the immune system was likened to armed forces, your Schutzwaffen, your defensive weaponry against the outside hostile world. We now are looking at the immune system more like bouncers or a passport, really mean passport control agent. They know who to let in and who to keep out. They are actually, the immune system is not just dumb soldiers killing everything that's foreign. They're discriminating between things they know to be useful and helpful and those which are known to be harmful. And so we have in the literature these crazy passages which would not have made sense when I was first teaching immunology. These results underscore the adaptive immune system's critical role in establishing a sustainable host microbial relationship. My soldiers are turning into sissies? I mean, what is this? These soldiers are now making sustainable host microbes relationships. This may involve the creation of an optimal symbiotic environment on the interior of the Peyer's patches. Peyer's patches are what makes IgA, which destroys, viruses. No, it's actually making an optimal symbiotic environment. Therefore, commensal bacteria exploit the toll-like receptor pathway to actively suppress immunity. Toll-like receptors activate immunity. We propose that the immune system can discriminate between pathogens in the microbiota to recognitions of symbiotic bacterial molecules in a progress that engenders commensal colonization. In other words, the immune system is actively helping the bacteria get a home in the body. This is a new role for the immune system. Not a new role evolutionarily, but a new role in our understanding. We did not know the immune system helped bacteria. Okay. But what we find is that the bacteria, the immune system, and even the diet are critically important. This was a paper that came out last year where they took macaque monkeys, rhesus monkeys, and they gave some of the newborns formula, which is nutritional baby formula, which would help them grow, and they let the other ones be fed with mother's milk. And what they found was that they got a different population. I mean, I'm talking different, non-overlapping populations of bacteria here. Not totally non-overlapping, but they could be diagnostically separated based on the bacterial population. And what they found was that those which were reared with the mother's milk had a population of bacteria that actually made Th17 cells, much more than those that were just given formula. And this was that the bacteria promoted by breast milk made arachidonic acid. Remember I said that the breast milk actually allows some bacteria to grow. The arachidonic acid induces Th17 cells. The Th17 cells are those against Candida and Salmonella and Staphylococcus. So they're against other bacteria. And the breastfed macaques have more Th17 cells to a very statistically significant degree than the formula-fed macaques. So there is this interesting dialectic here. The immune system, which regulates bacteria into the body, is being made in part by bacteria. Okay? So the bacteria and the immune system interact to form populations of bacteria to form the cells of the immune system. And this has been shown over and over again, that the cells of the immune system are in part made by signals from the bacteria. And that's what I'm going to just mention briefly here when we talk about developmental individuality. Now, this is an optical illusion. Can you see the fetus here? Here's the eyes. Here's the hand, the rump, the legs. Let's say that the fetus is partially constructed by the environment, that it is not something that comes directly from the zygote. And so we have here the lights, the Euprimna, the squid, is kind of the poster organism for this, that the squid, when it's born, does not have a light organ. The light organ is made when the ventral surface absorbs Vibrio fischeri bacteria. And the Vibrio and the squid collectively make the light organ that houses the bacteria, allows the bacteria to multiply like crazy, and allows the squid protection from predators. So it's an organ. The light organ is an organ made by the symbionts and the big guy together. The orchid. The beauty of the orchid is made possible by a fungus. That's why orchids only grow where they're fungi. That the orchid seeds are so small, so lacking in carbon resources, that they have to be opened up and given resources by a fungus hypha. So the orchids are beautiful examples of symbionts. Then wasps take over the symbiosis later. But Euprimna, the squid, these are beautiful examples that organs come into existence through symbiosis. And that happens in us mammals. The development of the immune system and the development of the gut capillaries depend on microbes. So this is the capillary system in green of a germ-free mouse. And it's rather poor capillary network. Intestinal vessels in a mouse without microbes. You add the microbes back, and if you do this within the first week, the intestinal villi get their circulation. So here we see a good circulatory system around the villi. This is important. These are the capillaries that get the nutrients out of the villi, out of the intestine. Similarly, with the immune system, here we see the dome and the follicle of the gut-associated lymphoid tissue, the activated B cells and the activated T cells of the immune system. If you don't have bacteria in them, you do not get the dome and follicle. You don't get gut-associated lymphoid tissue. There seem to be two bacteria whose products are critical to get the normal lymphoid tissue. In other words, we have subcontracted, outsourced, think of different metaphors, we've outsourced our developmental signals to the microbes. They're always going to be there. They're always expected to be there. Why not make them useful and have them do something to construct our bodies? So our bodies rely, in part, on signals from the microbes. How do they do this? Okay, this is a paper by Laura Hooper, and colleagues in Gurdon's lab, 2001. This is the paper that drew me into the field. This is the paper that, I just looked at and go, I'm supposed to be writing a book on developmental bio. I have left out a major component in leaving out the microbes. What this is saying is here is the relative mRNA abundance for 3 proteins. Colipase, angiogenin, it should be angiogenin 4, but angiogenin, SPRR2A. An enzyme that's involved in lipid metabolism and transport. This is a protein that's involved in forming blood capillaries. This is a protein that's involved in making a matrix. Here are the germ-free mice. This is set to 1.0, relative mRNA abundance. Here's your 100% mark, 1.0. Okay, that's germ-free mice. Here's conventionally raised mice. Oh, for colipase, it's 6-fold more expression. Angiogenin, 10-fold more expression than germ-free. And here we have 50-fold more expression than germ-free. This is what's normal. This is what you get without the microbes. And this is what you get by putting different microbes in. Bacteroides, this guy over here, is really important. Bacteroides makes the angiogenin, and it overshoots SPRR2A, has to be brought back by the other guys. This is what happens if you add gut bacteria to germ-free mice. You basically take it close to the baseline. So what this is saying is that the bacteria are inducing gene expression, normal gene expression, expected gene expression to make our bodies. Angiogenin is involved in making those capillaries. Now, angiogenin has an off-label use as well. Okay, Bacteroides induces angiogenin. Okay, that's over here. Okay. Angiogenin helps make the capillaries of our gut. That's good. Angiogenin also is bactericidal against Listeria, the major competitor of Bacteroides. So we're contract killers for Bacteroides. Bacteroides says, okay, I'll make your gut, but you know what? You help me too. You get rid of my competitors. We've not only co-evolved, we're co-developed. Much more intimate. Okay. Zebrafish, the stem cells divide under signals from the microbes. The microbes are actually causing the division of the epithelial stem cells in the gut. So this is even more important than the capillaries. It actually helps make the gut of the zebrafish. Really interesting stuff that's coming out, almost science fiction, that the material that the bacteria are producing may affect brain development, normal brain development. So here's NGF1A and BDNF. This is a transcription factor and a paracrine factor involved in plasticity. What you see is that the mice with the bacteria have much more gene expression than the mice without the bacteria. So this is your normal gene expression. This is your gene expression without the bacteria. Without bacteria is like being a mutant. It's like being like having only 12% of the normal mRNA for colipase, or 2% for SPRR2A. Bacteria are normal in development. This is some work that's been done recently that germ-free mice actually have an autism-like behavioral syndrome. They are involved in self-grooming, and they don't have exploratory behavior. This is, Bacteroides seems to be very important in this. And this is a very important paper from Sarkis Markesian's laboratory showing that microbiota modulate behavioral and physiological abnormalities associated with neurodevelopmental disorders. They can induce a mouse model of autism by stressing the mother mouse and stuff with viral challenge. Giving the mice huge amounts of viral challenge, make its immune system work overtime. The pups actually have these symptoms which look like autism. Whether or not it's autism, we don't know. But it's, again, exploratory behavioral acts, prefer solitude, excessive self-grooming. And what's interesting is that you find that there's a lot of leakage in the gut epithelium. If you add back Bacteroides to this is very lacking in Bacteroides. You add Bacteroides back to it, the adhesive membranes actually change and get more adhesive. There's less gut permeability, and certain chemicals that are made by the bacteria, which are not good chemicals, they can be poisonous and actually cause anxiety and stuff in mice. These chemicals are reduced when you add back the Bacteroides. So there's a whole new study here of what the relationship between normal brain development, normal synapse formation, normal synapse conductivity, and the gut microbes. There's something called the gut microbe-brain axis, which you can Google and read lots of papers on. Okay, last thing I want to talk about individuality is evolution. And Lynn Margulis, life did not take over the globe by combat, but by networking. And there are many mechanisms that symbionts can be involved in speciation. One I mentioned, genetic variation through symbionts, the P. aphid story I told, thermotolerance and color being transmitted through alleles of the symbionts. And there are others as well. There's also reproductive isolation through the symbionts, selective mating preferences that symbionts can give, and also cytoplasmic incompatibility, so that strains with different symbionts within the same species don't interbreed together. But one of the most interesting is the work, I think, interesting, coming out from Nicole King's lab at Berkeley, which says we may have never been individuals. Bacteria had a 2 billion years head start on eukaryotes. And that the whole formation of eukaryotes might have been to make more niches for the bacteria. That if you have certain choanoflagellates. Okay, choanoflagellates are the sister group to animals. Okay, so choanoflagellates are the protists that are most similar to animals. And they are protists. They're single-cell organisms, except if they're co-cultured with certain bacteria, in which they become multicellular organisms. They become a multicellular organism on the basis of connections between the cells and an extracellular envelope, a lamina, a basal lamina. So they become epithelial cells. So this is like your first step towards animal multicellularity. So again, they're normally single cells, but if you co-culture them with this Algoraphagus bacteria, which is a Bacteroides type bacteria, you culture them together, which they're often found together in nature, you get multicellularity. So we may have become multicellular through a symbiosis with bacteria. So there's really no individuality for animals. We're not anatomically Individuals, 9 out of every 10 cells of our body may be microbial. We're not physiologically individuals. We have joined metabolic pathways. Developmentally, certainly not. Gut microbes help build our gut, immune system, light organs. In certain insects, they prevent ovarian death. Immune individuals, the actual microbes help build the immune systems. They expand the immune repertoire. Genetic individuality, the genome evolved with those of symbionts. I haven't gone into the symbiont, the nucleus-symbiont interaction. We have over 100 different genomes in our bodies, and some in many insects have phenotypic outcomes. Evolutionarily, symbionts can provide selectable variation and isolation. So I would say that we are teams. Okay? So, if we are teams, as I said, I think we've been using the wrong view of the body. We've been using essentially a racially pure body. We've been using a body which every cell is the same. But we know that's not to be the case. So this talk is being given by Team Scott Gilbert. And I just want to say, what does this mean? Well, it can mean different things. And I want to go back to the body politic metaphor and also to what it means for biological sciences. What it means for biology is we have a whole new set of things to study. We didn't know diddly squat. We thought we knew basically, what biology was about. We now have a whole new layer to think about, also to think about during our experiments. When we control, we have genetically identical mice, and we give them identical food, but they do not have genetically identical bacteria. And so this is something we haven't accounted for. And the bacteria will metabolize drugs differently. It will metabolize cancer drugs differently. They'll metabolize endocrine disruptors differently. So we have all these questions that we have to ask now. How do the symbionts enter the body? What's permitting them? What signals are the symbionts using to form our capillaries and lymphoid tissue? All sorts of things. What's the evo-devo of holobionts? How do the parts get integrated to form a whole? So we have lots to study. For the social sciences, okay, going back to the body politic, and I just couldn't help say, what does this mean for immigration policy? Okay, if you have a monogenomic animal, the symbionts are gastarbeiten. They're the workers who are allowed in and then gotten rid of. They literally do the shit work. If we are culturally pure and we invite in this other species, then we get rid of them. Okay, if you have a model where the animal has more porous boundaries, again, talking boundaries here, well, then the bacteria are kind of lawful permanent residents. They have green cards. They're allowed in our body, and they can stay there. They might be under more surveillance than the other cells, but they can be there. But in the holobiont, they're full citizens. That the symbionts help build the animal and benefit from it. So just the language that we use to talk about symbionts is often a cultural language, and we have to be careful about that because it may reflect our views towards the polity itself. Philosophy. What is life if life is both an amalgam of becoming with the other, a little Deleuzian and Haraway philosophy here, as well as the Hobbesian battle of each against all? Usually when we talk about individuality, we're talking about the battle of each against all. I am an individual because I'm not one of you guys. I am able to find my resources and battle for them. But now we're talking about becoming with the other. Not metaphorically, but very literally. We become, we develop with the other being of a different kingdom of life. One of the metaphors that I'm using now, I'm playing with things right now, playing with the language that we use, is the notion of making the team. Okay? Those of you who've been in athletics know that making the team is a competitive, often vicious circumstance. And it is. When I say that Vibrio fischeri and the squid get together to make this light organ, that's because the squid poisons every other type of bacteria. And only Vibrio fischeri, which is a very small percentage of marine microbes, is able to stick. So there's competition to make the team. And it isn't only the best. Those of you who've read The Boys in the Boat, I think this is a wonderful example of the notion of making a team and then cooperating within the team to compete against other teams. And then the integration of teams into networks that are stable, cooperative structure that might compete with other leagues or ecosystems. You have here this amalgamation of competition and cooperation, which we really don't have a good vocabulary to discuss. And if you want to talk about this, please do, because I'm trying to find words to think about competition and cooperation. Okay, new view of health. Okay, just talk about these bacteria as being essential for our bodies to be built properly. They are also seem to be essential for our health, and we're at a point now where all sorts of associations are being made between microbe A and you need microbe A and if you don't have it, you'll get asthma. If you don't have it, you might get Parkinson's disease. If you don't have this one, you might get diabetes type 1. There are all these associations being made. Causally is the next step. We have to find out if they're causal. But last week came, or October 30th, the Unified Initiative to Harness. What a great metaphor. Earth's microbiomes. In other words, we're going to try to domesticate Earth's microbiomes. That is a big deal. They don't necessarily perhaps want to be domesticated, but we are going to try to harness them. And again, you think that the microbes are going to be medicine, they're going to be food, they're going to be fuel, they're going to be clothing. So this might be a very interesting metaphor. It might be a bit hubristic, but it's certainly something which is in the minds of many people. Okay. For health, certain microbes essential for preventing diabetes, asthma, autoimmune disease. This is something that's an ongoing project right now. So I want to say that symbiosis is the evolutionary strategy that supports life on Earth, whether it's the rhizobacterial legumes fixing nitrogen, mycorrhizal interactions allowing plant roots and seeds to get their nutrition, endophytic fungal protection against desiccation, the coral reefs and tidal seagrass ecosystems that sustain oceanic biodiversity. These are the huge symbioses that sustain the globe. And within these big symbioses are the smaller symbiotic webs that we call organisms, and the products of ancient symbioses that we call cells, and the products of even other ancient symbioses that we call genomes, that I haven't gone into the symbiotic relationship of genomes. Since this is about the boundaries of the human, and since part of the boundaries of the human are the animal kingdom, and the other part is technology, the cyborg notion, just want to show you these pictures. These are bacterial colonies on your cell phone. Okay? Put your cell phone on agar. Take it off. Grow the agar. This is what you get. And forensic biologists are saying they could tell different people apart. Personhood, identity, individuality. They could tell different people. They could tell who was using your cell phone. Okay, and this is kind of cell phone art. They're dirtier than toilet seats, by the way. Of course, no one cleans their cell phone, right? The two dirtiest things, cell phones and the bottom of your backpack. Okay, so we are all lichens. That's the point I want to bring is we are all symbionts. We're holobionts, that we cannot consider ourselves to be unigenomic organisms. We are multi-lineage organisms, which doesn't fit into the phylogenetic trees that you see in textbooks. That's because the phylogenetic tree is actually like a real tree full of symbionts. Thank you very much. Okay, be happy to try to answer. Well, this is small enough. You can just. Yes. Oh, I see. I see. Yeah, there's a.
Thanks for your talk, first of all. It's awesome.
Yeah. Tap it again. Yeah.
Okay. So thanks for your talk. So I wanted to ask, obviously, about individuality and push you on a point, because although I take it as a very successful attack on the notion of individuality, you still seem to be operating with some principle of individuation for the holobiont. So near the beginning, you talked about persistent microbial communities being incorporated into the holobiont. Then you seem to shift to talking about kind of function, so what aids in our developing our functions as part of the holobiont. And then you shifted into talking in terms of normalcy, or what's normal for a thing is what constitutes it. So those all sound to me like principles of individuation, how to individuate a holobiont on the basis of some criteria. So I just wanted to push you on that.
No, it's really a critical question because, and it's, I wish I was here to hear Hannah Landeker's talk, because you have something very similar in metabolism, where we keep our individuality only by changing our parts. And so where do you fit into this? You know, are we going through the world, or is the world going through us, or is it, you know, both? And I think that we're seeing something very similar now with the holobionts, is that we're keeping some sort of individuality by having the holobionts be part of us and metabolize in us and become literally an integral part of what we would call an organism. But they're not, you know, they can leave, and they do every day. So this is, I think, one of the tensions. And we were talking about this earlier with the whole notion of fertilization. In that fertilization, you know, we're talking primal here, that you have an egg, and the sperm enters it, and what's the first thing that happens is that the blocks to polyspermy occur to say, okay, a sperm has entered, no other sperm is going to enter. And so it's basically, it's separated itself from the environment in a way it never needed to be separated before. So I guess what I'm saying is that we are individuals, we're holobiont individuals, but we have a notion of individuality which is needed, if for nothing else, for interaction. Because you can't interact with sameness. You have to interact with difference. And so, again, these are things I'm playing with now, and other people are playing with, is what is a holobiont individuality? What does it consist of? Is it evolutionarily a level that can be selected? I think it can be, and I'm working with people, you know, who are interested in this question of levels of, you know, of selection. But I think you're asking the right question. Where are the boundaries of this new holobiont? And I think that they are much more porous. I think that they are, that with each meal we take, our holobiontness changes. We get different we're selecting different populations of bacteria. So it's a whole new way of thinking about individuality, which I think relates to Landeker's notions of metabolic individuality. Yeah. Oh, you're welcome.
That was so much fun. Can I take you in a slightly different direction? Which is
Do I have a choice?
No, you have no choice. Which is to talk about, you were saying you're trying with metaphors, you're dealing with metaphors. And so I wanted to offer, is it not on? Oh. I want to talk about the metaphor that I hear, or the association I hear in your title. We are all lichens. We have never been individuals in your things. And that makes me think of Bruno Latour, and we've never been modern. And in Bruno Latour, the whole point is modernity is the suppression of monstrous differences. And I wondered if you're working with that, and if you are, then whether there isn't a sort of question of having to enroll allies for any particular argument you're making by hitting at a scale that will speak to those allies.
Yes.
If that makes sense.
The answer is yes, we made our title as a kind of pun on Latour. And the notion of making alliances is absolutely critical. This is critical to the whole kind of theory of life, is what alliances are you making and with whom or what. And this is something I'm trying to take in other directions sociologically. For instance, alliances of science and religion. For instance, where, and an alliance, what I like about alliance is that it doesn't mean a takeover. And it doesn't mean a subsuming of one by the other. It doesn't even mean that it's permanent. And I use an example, the Grand Alliance of World War II. The leading communist country, the leading feudal country, the leading capitalist country, all in alliance for a common cause. And, of course, it broke up immediately afterwards. And so the alliances don't have to be permanent. They could be for a particular cause or for a particular moment. And so, actually, alliance politics is what I think is happening in the immune system. And I'm actually talking with Fred Tauber, who's an immunologist and a philosopher of biology, about the immune system's role in basically mediating alliances with microbes. Yeah, it's exactly the type of thing we're trying to play with.
Thank you so much for this very engaging talk. I am going to have to look at my dirty phone because I've just finished a box of antibiotics, and without my bacteria, I don't feel completely myself, which actually...
Literally.
Literally. But this is actually connected to my question, because within one of your first slides, and interestingly, you sort of did come back to him or them later, but I immediately started thinking of Deleuze and Guattari. And I do need to qualify that I'm in no way challenging any of the science or any of the scientific data, but I am going to be asking questions that your presentation made me think about, social things, or like from a social point of view. And I guess with that example of the woman, the pregnant woman, the bacteria, and that sort of dialogue. Mm-hmm. So the first question that comes to my mind is, where is the intelligence of the body and where is the agency? And within that, as you went on, I was thinking, it's much easier to think of a rhizomatic structure as Deleuze offers, but that structure doesn't work if we employ kind of, I guess, like this model of we're not individuals, because in that structure we are individuals, but we're connected, we are within a network. Mm-hmm. And maybe the bacteria are obviously extensions of my body, they are within this body, they are within this island. And, so I guess I'm kind of wondering like where you think the intelligence and the agency lies, because maybe the woman's body at the third trimester needs to slow down, and it needs to take better care of itself, and it needs to do something to produce that bacteria so that it can induce all these effects. So maybe there's some kind of intelligence there. And I feel like, there are bodily ways of knowing things too. So it is more than just the bacteria that affects genes and DNA. There's behavior, environment. There's a lot of research that says that babies who are being born now, they are much better able to, use the phones and everything, and that's definitely something being passed on after generations of people using these things. And that's not necessarily, ancestral genetics, but it's, it gets in the body, and it's,
I don't know about the phone system, but certainly there's really a lot of interesting things about what the body is knowing. And I would say that, when you say where is it, I don't think it's a locus. I think it's relational. I think that the intelligence, whatever that might be, and I don't know what it is in the relationships. And it's interactions of things which are, a very diffuse locus, if you would. But I don't think that, when you say, I don't know where there would be a locus of intelligence. I think that the relationship becomes this, very robust intelligence, if you will, because it's meeting it's meeting challenges on all sides, and it tries to keep itself constant, which means it has to call in alliances, it has to have redundancies. So it's this network of interactions, which maybe one could call an intelligence. I don't know. But I think that, that it isn't something that's going to be localized. I think it's a very diffuse entity, whatever this.
If I can quickly add,
yes.
You gave the example of, because that was really wonderful, how you were making this analogy between, social engagement and the body, that metaphor. And I thought if all of the illegal immigrants in New York today didn't turn up, none of the restaurants would work, nothing could function. And just this idea that, you probably know better, but recently there's some research that says that like people who have cancer don't get sick, like they don't get high fever, and they're now looking at new ways of healing cancer by maybe, doing that. So yes, the body has an intelligence, and it's maybe not localized, but where is the agency? Because the moment we say, we built that hierarchy, you talked about the orchid, Deleuze also talks about the orchid, that it's a together sort of system. So maybe we exist so the bacteria have a place to live, right? So, I mean, that's kind of what I'm asking, but...
Yeah, I think it's, kind of dialectics all the way down, that it's I don't think that it's going to be possible to find a site of agency, but I think the relationship itself may be the agency that we're looking for. And as I said, it's something which is generated, it comes into being, and that nothing by itself kind of has an essence, but only in the context of where it is at that particular time and place. It's a rather Deleuzian Haraway type of notion that the smallest unit, if you will, of analysis is, the relationship. But I don't think you can get away from that. I think that that's where the agency is going to be, in the relationship itself.
Thanks, Scott. That was a wonderful talk. And obviously it resonates with all the work we're doing in the lab. So we're very happy to have met you personally. So my question relates to population genetics theory, because I think you find very strong opponents of these type of thinking, given the theoretical framework that we've been working under for the past century, and the synthesis, and when Seth Borderstein has started to present his perspective on symbiosis and how evolution can shape, how symbiosis can shape evolution of organisms, and shifting how we think in terms of units of selection. And there is this just pushback by Torelli and probably people like Michael Lynch and, these hardcore population geneticists that are the ones that have been dealing with the theory of evolutionary biology. How is there going to be a middle ground, and where is it going to come from, and how is the theory going to be shaped by these avalanche of people interested in symbiosis that are primarily empiricists and not theoretical biologists? Because Ivo Divo got the same kind of pushback when the tools became available and people started to just say, well, let's think about this differently and think about macroevolutionary perspectives differently. But I feel that that's going to be, people are entrenched in their corner, and they have the theoretical tools to argue very strongly against proponents of these new ideas. So since you are clearly embedded in the debate and discussion, I'd like to hear your perspective of where can we go from here.
Okay. Yeah, you're absolutely right. You've seen the debate in Nature that does evolutionary theory need a rethink? There's a debate, I think, about a year ago. It was called, Does evolutionary theory need a rethink? And there were pros and cons. And the pros who said definitely yes were mostly people like Lalland and people who were working on niche construction. And the people who said yes, said no, it does not need a rethink, were really interesting. They included people like Hopi Hoekstra, and they included people like Greg Ray. Now, these people are looking at alleles of developmental regulatory genes. So they are incorporating that old, the old bête noire, the old devil of evo-devo. They're saying, oh, we could tame that evo-devo. We're going to put it in population genetics. And they're doing that. And it's really interesting. And I think this is exactly what they should be doing. They should be looking at allelic differences of genetic regulatory genes. I think that that's a way of getting at intraspecific variation. What they were arguing against was the eco-devo, was the symbiosis and plasticity. And I think that to them, to the mathematical population biologists who grew up under the notion, Dobzhansky said it specifically, evolution is a subset of population genetics. And Haldane said you do not need to know about development to understand evolution. So these were very explicit things said by the masters. And I think that to those people, what I just said right now is natural history. It's not evolutionary biology. It's not even within the domains of any particular cell biology or molecular biology. It's natural history. So we're embarking on some projects. And this was done at a meeting by Snate Gessis and Eva Jablonka last year. They got a bunch of evo-devo people, eco-devo people, evolutionary biologists together. And so, believe it or not, I'm working with Lisa Lloyd, and I'm working with Joan Roughgarden, who are real evolutionary theorists, game theory and selection theory, and we're talking. And the hope is we're going to have, for this volume, we're going to have separate chapters. And also the Rosenbergs are involved in this, too, Alana and Eugene Rosenberg. And so we're talking to each other right now. And the hope is that after we make our separate publications, if there is overlap and if we feel it works, to try to make a joint paper, taking this natural history and putting it into the framework that evolutionary biologists respect. And it's a matter of respect. I don't think they respect this. So that's, I think, where we're at right now. And I'm not the only one doing this. There are Thomas Prudhoe in Paris, Bordeaux is doing this. The people in Tel Aviv are doing it. There are a bunch of people who are trying to get the evolutionary biologists interested in this question and not as a knee-jerk response saying, no, we know it's wrong. But certainly, population genetics is something that emerges within a species. And that was Thomas Hunt Morgan, 1926. He says the only way of studying evolution scientifically is to study it by genetics. And that meant intra-species. And so all of the population genetics is really done within a species. It's not going to tell us how you get new species necessarily. It totally ignores symbiosis, which, as we know, is a source of variation and a source of inheritance. So it's going to take a while. As you mentioned, evo-devo, it took about 30 years for the people to agree that evo-devo is part of the evolutionary biology. And I figure evolutionary biology kind of grows like a pearl. You have the Darwinian Wallace framework, and then you have Fisher and Haldane and Dobzhansky and Hamilton, and now the evo-devo, and what's waiting on the outside to be put onto the pearl is eco-devo. And that's going to take a while because it's non-genomic. Yeah.
I want to go back to metaphor for a moment, if I may.
Yeah.
I'm going to say this as the arts and humanities person, because what I'm interested in is partly, well, we get the question every once in a while, why is the Institute for the Arts and Humanities doing this thing that's straight up biology? And my standard answer is what we're talking about, what it means to be human and the meaning of life, literally. Literally. Mm-hmm. And so what fascinates me most about this is not the analogical or metaphorical relation to the body politic, although that was fun. I think back to another relation, back when humanists were trying to understand physicists. And we said, oh, yeah, quantum theory, uncertainty principle, yeah, that has implications for the way we investigate ourselves because we can't basically know both our speeds and our positions at the same time. No, that doesn't really work. Right? The relation was only one of analogy. Mm-hmm. And it seems to me that if the relation, if you take, nations are like bodies, and that therefore your view of symbiosis has implications for immigration and what have you, that is just metaphorical or an analogical relation. I find what's more productive really is the literal.
Is
The
it
literal,
homologous?
that this has implications quite apart from any policy, any guest worker program, anything, that understanding this on the level of the molecular biochemistry itself should have implications for the arts and humanities, quite apart from any analogy. So that's what I'm really most curious about, is what, to take an analogy actually from the work of Catherine Malibu, and it has implications for epigenetics. The idea that, traumatic historical events are so traumatic that it's almost as if they leave lesions in your brain. No, they really do. Mm-hmm. No, those are literal lesions, right? And so if you could just dwell on that part of your talk toward the end, where you say, I'm playing with this because we're not really sure how to speak about this yet. How much of that playing involves literally, the emphasis not on its implications, for social policy, but the literal philosophical reorientation it takes to conceive of life in these terms?
Yeah. No, I think that we have got to go, a massive readjustment in our thinking. I think that we think of ourselves, in a way that has been, I think, wrong. You could say it's oversimplistic. No, I think it's more than oversimplistic. I think it's fundamentally flawed and wrong. So what do we do? And we have to go through the stages of relearning. And I think that we have to relearn biology in terms of alliances and relationships. I think we have to relearn anatomy in terms of what are the symbionts' contributions. It's so weird. It's almost like, feminist theory of the 1970s. So these guys have been put in the margins. We haven't talked about them. No, they are critically important. It's taking what was marginal and making it central. And I think that, is so, important to what we think biology is. I think it's important to what we think health is. I think it's important to what, we're talking about alliances. How does the body make alliances? How does it make alliances with things which want to multiply and perhaps destroy the body? How does it keep it under control? How do they interact with each other? I think there's a lot that we can learn from the body. The body is a polity, and it solved a lot of problems of differentiation, of integration, of interrelationship. How does the body do this? I'm interested in knowing that. I don't know if this will be, in biology there's a split between something being analogous and something being homologous. Is it coming from the same source and manifesting itself in different ways? Or are they just two ways that come together to look the same? And I don't know which it is, in the body and the politics. I don't know if there's a way of organizing that is common to these things, like natural selection is. Natural selection's incredible. It works on automobiles, it works on bicycles, it works on humans, it works on elephants, it works on anything that multiplies with variation. It's incredible. That is, something that you could say, that evolutionary processes kind of work, selectively, across wide realms. I don't know if these organization principles, whatever they might be, are going to do that. But we've just realized that they're there, and that these that we are multi-lineage, organisms. That's the term, multi-lineage organisms. That's not something that was in. I don't think it's still in the textbooks. I don't think it's there yet in the intro bio books. I should take a look. But yeah, it's literally a new world out there. And I think just like the microscope, opened up a whole new world of new beings, what PCR and high-throughput RNA analysis, which is, the most reductionist thing you can get, what they've discovered is a world of relationships, which we had no idea existed before. So, this is all new. To me, it's incredibly exciting. I think to some it's, kind of very frightening. But I think that this opens up opportunities to not only study the world, but to better our health and stuff, which are just like the microbial revolution, in the 1880s. So it's going to be fun.
And thank you again.
Is it? Yeah.
I just have a quick question.
Yeah.
It's really more a matter of, thanks for this. This is immensely helpful, and I'm not a scientist. I'm more in the sort of humanities end of things. But I wonder about the insistence on what happens to the organism in all this, because the organism still seems to be the kind of linchpin figure to some degree. So you've nicely deconstructed how the individual works, but it does seem like the organism remains the kind of tactic by which everything else. So when you talk about relations, you seem to be talking about relations between pre-existing things. So we're born as an island, and then we're colonized by the bacteria. Okay. I mean, I wonder if you push this far enough, isn't it just there is this sort of microbial sort of biome, and there are various sort of phase transitions on this biome that make various kinds of layerings that make these things that we call organisms. Or is it that the organisms really become the sort of, it's like relations between pre-existing things, or the things are only more or less accretions of the of the biome itself, which would seem to me to slightly be two different things, or maybe not. I don't.
Well, the notion of codependent construction, going back to Nagarjuna, when we're going to this notion of that nothing exists in and of itself, and the concretion of processes, Whiteheadian processes, makes what we consider to be individuals. And so everything really, when you said that things are interactions of pre-existing things, and those pre-existing things, of course, were from interactions of other, and so you, as I said, dialectics all the way down. I think that, from a microbial point of view, we are just niches. And you can get into all sorts of, this is where I think that Dawkins' notion of extended phenotype gets into absurd things. Because if we're the extended phenotype of a bacteria, all the different bacteria have their extended phenotypes in us. We're niches for the existence. So does that mean that the bacteria are propelling us to multiply? To make more niches? We get into other questions involving the needs of bacteria. But I think that, you're right in the sense that we come into existence through the interactions of pre-existing things that came into existence through the actions of other pre-existing things. Okay. Turtles, I study turtles. That's my research. All the way down. Thank you.